The C peptide is not present in the mature insulin and it is removed during the maturation of the insulin. If ADA is only not synthesized properly, then how will the immune cells work, right? So that is what happens in the case of people who are deficient of ADA. So gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or a embryo. Hello everyone, a warm welcome to another session on chapter 12 of second PUC, that is biotechnology and its applications. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the role of biotechnology in the field of agriculture, wherein we learned how green revolution was adopted in order to increase the yield of the crop, and also we studied about two important genetically modified crop, that was BT cotton, how it was genetically modified, and also we learned about the genetically modified tobacco plant as well. In today's session, let us study about the role of biotechnology in pharmaceutical industries. So, let's start with studying about the biotechnological applications in medicine. So, the recombinant DNA technology processes. So, when we say recombinant DNA technological processes, so about this we had discussed in the previous chapter, right, wherein the gene or the sequence carrying the genes of our interest are isolated from the organism, purified, they are joined together using a particular enzyme, and then these sequences are sent through the vector, it is sent into a host organism. So that particular host organism will start developing the characteristics that we want or we desire, right? So that is what is exactly done in recombinant DNA technology. How it is done in a long process, if you want to know about it, you need to visit the previous sessions of the last chapter. So the recombinant DNA technological processes has made immense impact in the area of healthcare by mainly enabling mass production of safe and more effective therapeutic drugs. So the best example we will learn in today's session is how exactly insulin is manufactured, because insulin, to manufacture insulin, there are millions of people in a country who are diabetics and who are insulin dependent. So to fulfill their demand for this particular medicine, insulin, a large production of insulin is needed. So how exactly can insulin be produced in a larger extent within a short span of time? We shall see. So when we talk about that, we will understand it better. So therefore, under biotechnology, there is one technique called as recombinant DNA technology, which is mainly used in order to produce more effective therapeutic drugs. And further, the recombinant therapeutics, they do not induce unwanted immunological responses because they are directly created by using genes or by altering the genes. Therefore, the chances of any side effects or the unwanted immunological responses are quite negligible in the case of those therapeutic drugs which have been produced by recombinant DNA technology. And also here, the recombinant therapeutics do not induce unwanted immunological responses as when compared to those products isolated from non-human sources. So when I say non-human sources, I'll give you an example in the coming slide, wherein insulin earlier was extracted from pig, but it created a lot of problems. So pig is a non-human source, right? Does pig come under the human family? No, it's a non-human source. But when insulin was isolated from pig and administered to human beings, it had a lot of side effects. So that was one of the reasons why that particular procedure was abandoned. But that is not the case in recombinant DNA technology. In recombinant DNA technology, wherein recombinant therapeutic drugs are created, those drugs will have very, very less side effects, or no side effects at all, or no harmful immunological responses or unwanted immunological responses when compared to those drugs which have been isolated or which have been prepared by using non-human sources. So, at present about 30 recombinant therapeutics have been approved for human use throughout the world. So, there are about 30 recombinant therapeutics, that is using recombinant DNA technology. 30 therapeutic drugs have been prepared and it is being used throughout all over the world. And in India, 12 of these are presently being marketed. Out of the 30, 12 of these are marketed and insulin is one among them. Because they have given insulin as an example, I will be using the same example throughout this particular session. So, next let us talk about the genetically engineered insulin. So, I told you the insulin which was extracted earlier from pigs. But what happened when it was extracted from pigs and administered to humans, we shall look into. So, management of adult onset of diabetes is possible by taking insulin at regular time intervals. So, it's not that one tablet you swallow for a day and it is done. No. Whoever is under insulin treatment, the diabetic patients who are insulin dependent, at given intervals of time, they need to take insulin shots. So, imagine so much amount of insulin to different insulin dependent patients throughout the world. How can a particular company provide it? They can provide it by genetically engineering insulin. How is it done? We shall look into. So, insulin used for diabetes in olden days, it was extracted from the pancreas of slaughtered cattle and pigs. So, earlier days insulin was extracted from the pancreas of the, because pancreas is the main organ, be it a human body or an animal body, pancreas is the organ that is responsible for the production of insulin, and that insulin was isolated from the pancreas of the pigs and the cattle during the olden times. But insulin from an animal source caused, in some patients, it caused to develop allergy or other types of reactions to the foreign protein. That's true, right? Because it is a foreign protein. And foreign protein, you have studied about this in antigen antibody reaction, right? How our antibodies will fight against the antigens which are the foreign sources because they do not belong to our body. Of course, even we also produce insulin, but saying that, can you take insulin from a cattle or a pig and then put it into the human body? Though it might have the same chemical structure, composition and all that, no, that is not possible. So, the same thing happened here. Now, the insulin that was taken from the pancreas of the cattle and the pig, that was used as a therapeutic drug to treat insulin dependent diabetic patients. But what happened was, some of the patients started to develop adverse effects. Why? Because our own antibodies will start to think that they are foreign substances and it will start attacking them, causing a lot of ill effects in the human body. So, insulin usually it consists of, so that is when they stopped using this particular method of insulin extraction. So, what is insulin? What is it exactly made up of? Let's look into. So, insulin consists of two short polypeptide chains. Polypeptide means having many peptide bonds there. So, it is made up of two short chains, that is chain A and chain B. If you can see here, insulin, it is made up of two short chains, that is one is chain A and chain B. So, we have chain A here and chain B. So, two short chains it is made up of. So, this two short chains are linked together by disulfide bridges. So, if you can see here, they are linked by disulfides, right? So, these two short chains, disulfide, because two sulfides are there. So, they are linked by disulfide bridges like this. So, these are the disulfide bridges. It is very important for us to know the structure of insulin, because to understand how it is genetically engineered, knowing the structure of insulin is very, very important. So insulin is made up of two short strands, chain A and chain B. These two chains are held together by two disulfide bonds. And in mammals, that is including human beings, insulin is synthesized as a prohormone. So prohormone, what is a prohormone? So prohormone is a hormone which remains as such before it is processed. Means before it gets separated, before the unwanted sequences and all are removed, before that what hormone exists, that is called as the prohormone. So in mammals, including humans, insulin is synthesized as the prohormone. So this insulin is a prohormone. That is why it is called as a proinsulin. Now here, why it is a prohormone? What did I tell you? The insulin is made up of two short strands, right? That is chain A and chain B, which are held together. The green one is the chain B, the blue one is the chain A, which are held together by disulfide bonds. But can you see here in between there is a long chain that is present here, which is a free C-peptide chain. This did not get removed here, right? It is there in the insulin molecule, as such it is there. So therefore this is a proinsulin. It is a prohormone. Now I hope you have understood what a prohormone is. Now this is not a prohormone. Why it is not a prohormone? Because this particular chain has been removed, and those chain that is particularly responsible for the production of insulin has been retained. So that is the difference between a prohormone and a hormone. So in mammals, first the insulin, that is even in human beings also, first the insulin is usually synthesized like a prohormone, and this prohormone, why? Because it contains an extra stretch of chain which is called as the C-peptide chain, as I have shown there. So this C-peptide chain is not present in the mature insulin. So first insulin is in the form of a prohormone. That insulin should get, if it has to produce insulin, that is proper insulin, it has to mature. If it has to mature, this particular free C-peptide chain should get removed. So this is the C-peptide chain or the C chain. This should get removed. So once it gets removed, then the prohormone forms a hormone, or it forms a mature hormone. So this C-peptide is not present in the mature insulin, and it is removed during the maturation of the insulin. And the main challenge for production of insulin using recombinant DNA technique was getting insulin assembled into a matured form. That was the main challenge. How to get insulin assembled into the matured form? Obviously, do you think that insulin is simply some kind of powder that is there in the pancreas? No, never think like that. What is that? It is actually produced by a stretch of DNA strand that is there in the pancreas cells, right? In that stretch of DNA strand, what all are there? There is A chain present, and there is present, A chain will have some sequence, there is A chain present, there is C-peptide present, and there is B chain present. Now that is immature insulin. That immature insulin should become a mature insulin. How will it become a mature insulin? The C-peptide should get removed, and only the A chain and the B chain, which are held by the disulfide bond, should get retained. So that will become a mature insulin, and that strand alone will produce the insulin required for the human body, and that will naturally occur in the human body. But if we need to create such a chain in the laboratory condition using recombinant DNA technology, then it is a very quite difficult process. So therefore, in 1983, one company which is called as Eli Lilly, it is an American company. This American company was the first company to produce the DNA sequence which corresponds to A and B chains of human insulin, and then that is, they were the ones for the first time, Eli Lilly company, which is an American company, they were the first time to separate the A and B chain, which are held together by disulfide bonds, and they put it into plasmid. Why should they put it into plasmid? Obviously, because plasmid, that is the E. coli, Escherichia coli bacteria, is the one which will clone this particular mature insulin, right? So that is why they put it into the plasmid. So the A and B chains of human insulin was taken, was prepared. Now, so now always remember it's only the A and B chain. Now why? Because it is a mature insulin. What did Eli Lilly company do? They designed a technique of recombinant DNA technology wherein they could remove the C peptide chain of insulin and only retain the A and B chain, which are held together by disulfide bonds, wherein the insulin became a mature insulin. That mature insulin they introduced into the plasmids of E. coli. So therefore, this E. coli could produce chains of A and B strands of insulin. So chains A and B were produced separately and they were extracted and then they were combined by creating disulfide bonds to form human insulin. So that is how they did. They extracted it from the humans itself and they delivered it to the humans. Now, the organism from which it was extracted is also human. The host is also the human. Which was the vector that was used? The E. coli plasmid vector was used. Now you understood, right? Earlier what they used to do? They used to take it from the pig and sheep and then put it into the human. But it caused a lot of side effects. So that is when they went in for this recombinant DNA technology wherein they could genetically engineer insulin. What they did? They isolated the strand responsible for producing insulin from the human pancreas itself. Then that insulin, first it is in the immature form. It is a pro-hormone or it is a pro-insulin. Why it is a pro-insulin? Because it contains a chain, C peptide chain, and also it contains the B chain, right? Wherein the A and B chains are held together by the disulfide bonds. So that was removed. So that is normally produced in the human body. So now that should be manufactured on a larger scale. So for that what they did? They isolated that. They went in for the recombinant DNA technology wherein they tried to cut the C peptide bond. The C peptide chain was removed. What two chains were remaining? Only the chain A and chain B of insulin was remaining. Now they subjected the chain A and chain B, which are separate. They subjected it to, in the laboratory condition, they created disulfide bonds between them, and that now insulin having chain A and chain B held together by disulfide bond is a mature insulin. That mature insulin, they put it into the E. coli bacteria and that is how they multiplied it, forming copies of it. Now copies or large copies of this particular mature insulin they got, that can be used for various pharmaceutical industry for the manufacture of insulin, or it can be administered into the human body. That is how this was done. So next let's talk about gene therapy. So how interesting, right? So imagine that is how medicines are prepared nowadays, through genetic modification. That is using the humans itself, the genes taken from the humans itself. One of the best example that we took here was insulin here. So what is gene therapy? Let's look into. So because gene therapy is one of the most advanced technique that is used nowadays in the medical field. So gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or a embryo. So this can be done. So when the fetus is in the mother's womb or when the embryo is in the mother's womb, after certain time period of month, they will have to undergo the chromosomal test, right? Wherein is there any defect in the chromosome? Will the baby have Down syndrome? Will the baby have Klinefelter syndrome? All that they should check, right? So if there is any alteration in the chromosome number or any alteration in the gene, that can be found out at that stage itself and it can be corrected. That particular procedure is called as gene therapy, wherein the genetic mutation in the particular child or the embryo, it's detected in an early stage and it is corrected. That is gene therapy. Here genes are inserted into a person's cells and tissues to treat a disease, or if it is an adult human being, the genes are directly inserted into the cells. So that gene will get incorporated in the DNA along with the DNA strand that is there, and it will start producing that particular protein or compound which can cure that particular disease. So that is also done in gene therapy. And correction of a genetic defect involves delivery of a normal gene. So here what they do is they deliver a normal gene into an individual or an embryo, so that gene, say for example, I don't know if this is true or not, but to make you understand, I'll just take an example. Say for example, there is no development of any hair, be it the eyebrow or anything in a particular human being because of some reason. Maybe keratin. Okay, there is a gene that is become defective, and therefore keratin is not produced. Therefore, the person's hair will be lost very fast. It will be very scanty, very thin. The nails also might not develop properly and all that. So now we need to correct that defective gene that was responsible for producing keratin. So that defective gene will be corrected. How will that defective gene be corrected? So that gene, whatever sequence it has, that same sequence should be sent. That is, a corrected sequence should be sent into the human. That is, it will be administered into the human body. That gene will go and sit in that particular defective gene's place, and it will start performing the actual function that it has to do. So therefore, it will compensate for the non-functional gene. Therefore, that particular disease or whatever is there in that particular human body can be corrected. So that is how it is done in gene therapy. So in simple words, it is nothing but whatever default is there in the genes leading to a different genetic disorder or different diseases, that can be corrected by sending a good sequence or a good gene, or that is same gene which produces the same kind of protein. So the first clinical gene therapy was done in 1990, and it was done to a 4-year-old girl. So this 4-year-old girl, she was suffering from a disease which is called as ADA, that is adenosine deaminase deficiency disease, that is ADA deficiency disease. ADA deficiency disease, it will completely reduce the immune system of a person's body, because adenosine deaminase is one such enzyme that is very, very important for the proper function of the entire immune system of a body. Now, if ADA is only not synthesized properly, then how will the immune cells work, right? So that is what happens in the case of people who are deficient of ADA. So the disorder is usually caused because of the deletion of the gene for adenosine deaminase. So in the stretch of DNA strand in the human body, there is one gene that is there, that is ADA gene, which is responsible for producing an enzyme, adenosine deaminase enzyme. This adenosine deaminase enzyme should be produced at the right time, at the correct amount, if the immune system of a person has to work properly. But in people who do not have this particular gene, or means there is complete deletion, or who do not at all have this particular gene which produces adenosine deaminase enzyme, they will be deficient of this particular adenosine deaminase. So if they are deficient, their complete body's immune system will come down. So in some children, ADA deficiency can be cured by bone marrow transplantation. So wherein bone marrow is responsible for the production of red blood cells, right? So therefore, if we transplant the bone marrow, wherein that bone marrow will contain healthy red blood cells that is capable of producing the adenosine deaminase, then the particular disease can be cured. But otherwise, it can be also cured by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But this procedure should be done every time, that is whatever time the doctor prescribes, that much time the ADA injections has to be administered into the body. Then the person can be cured of the disease. Not cured, but it can be controlled. But the problem with both of the approaches, that is the problem with bone marrow transplantation and the problem with ADA injection therapy, is that they are not completely curative. The disease will not get cured. Only thing, to some extent, the person's immune system can be triggered. Apart from that, the disease will not at all get cured with these things. So that is when they can go in for gene therapy to cure ADA. So what they did was, as a first step towards gene therapy, they isolated the lymphocytes. That is, lymphocytes from the blood of the patient was grown in a culture outside the body. So, at first step towards gene therapy, they took lymphocytes. So, what are these lymphocytes? They are part of the blood, right? So, they are some of the blood cells called as the lymphocytes, which are responsible for triggering the immune response in the body. You have already studied about the B lymphocytes and T lymphocytes, which actually helps in fighting. They are the antibodies which help in fighting against various diseases. So these lymphocytes were extracted. From whom it was extracted? It was extracted from those people who were suffering from ADA deficiency disorder. It was extracted and it was cultured in a suitable media in the laboratory. And a functional ADA cDNA, that is a complementary DNA, was introduced into that. So complementary DNA, how did they introduce? So first what they did, there is a ADA patient here. From that ADA patient, they took the lymphocytes. Now in that lymphocytes, is ADA being produced? No, right? It is deficient of ADA. That ADA, they put it in the media. And in that same media, they put a vector. Which vector they used? They used a retroviral vector here. So that retroviral vector is the one that is capable of entering into the lymphocytes. That is why they preferred a retroviral vector. So they used a retroviral vector, but that retroviral vector is not capable of causing infection. So that is how they designed that particular vector. So that vector and this particular lymphocytes, they cultured in one single media plate. And what they did was, into that retroviral, they added a cDNA, that is a complementary DNA strand that was capable of producing the ADA hormone or the ADA enzyme. So now that vector carried the ADA gene into the lymphocyte. Now what is there in the lymphocyte? The ADA gene is there. Now the lymphocyte was again put back into the patient's body. Now the lymphocyte automatically, because it is having the cDNA that is responsible for producing the ADA, the patient will automatically start producing the ADA. That is how they went in for gene therapy. Now the gene got corrected, right? In this patient, there was no gene at all that was responsible for ADA, but using gene therapy, they tried to introduce the gene through lymphocytes using retroviral vector. They tried to introduce that into the patient's body, wherein the patient continuously, he could produce the adenosine deaminase enzyme, and therefore his immune system improved drastically. And however, as these cells are not immortal, the patient requires periodic infusion. Again here there is a con, that is, there is some problem with this. What is the problem is that lymphocytes do not, so when you are born, do you think that the same lymphocyte cells will be throughout until you die? No. Cells will die, new cells will get born. So that is how it is a body, right? The old cells die and the new, in that place, new cells will get born. So those lymphocytes which were administered, which were corrected and administered, they will die eventually. Now, will the new lymphocytes carry that gene? No, right? Because it is a genetic disorder the person is having. So again, he will have to go in after some period of time, he will have to go for gene therapy. That is one problem here. But the thing is, however, if the gene isolated from bone marrow cells producing ADA is introduced into the cells at an early embryonic stage, it could be a permanent cure. So that is why when the fetus is in the embryonic stage itself, this procedure has to be carried. If it is done in adults, then the adult patient will have to go in for this therapy every now and then. But if it is done in the embryonic stage, it will get cured and it will be corrected permanently. So that is how gene therapy is used as a cure for ADA deficiency. So those previous methods that were there, they are temporary. You cannot cure them, but only thing is, at time intervals, the ADA can be produced. But this is a permanent cure. Permanent cure only if it is done in the embryonic stage. In the adult stage, it is not a permanent cure, because every now and then he'll have to go in for the gene therapy again and again. So therefore, it is better to get it done in the embryonic stage itself. So that is why nowadays a lot of procedures or neonatal tests are there which can be done in order to detect the chromosomal disorders in the growing fetus. So next, let's see how biotechnology is used in molecular diagnosis. So molecular diagnosis means the detection of various diseases. So how biotechnology is used to detect various diseases. So for effective treatment of a disease, early diagnosis of the disease and the understanding of that particular disease, that is the reason behind the disease, is very important. So using conventional method of diagnosis such as serum and urine analysis, all these are conventional methods, right? What do they do in serum and urine analysis? They will take the blood sample, they will take the urine sample, and normal culture tests they will do. But that results cannot be accurate most of the time. So therefore, instead of using the conventional methods of diagnosis, wherein early detection is not possible, we can go in for the molecular diagnosis mechanism, wherein recombinant DNA technology can be used and polymerase chain reaction can be used and enzyme linked immunosorbent assay, which is called as ELISA. If you remember, ELISA is one such assay to detect AIDS, right? But AIDS, if you just go in for this conventional methods, it is very difficult to detect. But with ELISA, it can be detected very fast and accurately. So these are some of the techniques that serve the purpose of early diagnosis. So what it means is that instead of depending on the conventional methods of diagnosis such as blood test and urine test and all that, which will delay the results, it is better to go in for molecular diagnosis techniques such as recombinant DNA technology, PCR and ELISA, which will give us accurate results at a quick span of time. So first, let's study about each of these molecular diagnosis tools. The first one is polymerase chain reaction. So polymerase chain reaction, in the previous chapter we had studied, right? Polymerase chain reaction, it is a machine which is used to amplify the DNA or make the number of copies of or clones of the DNA. So here, the presence of a pathogen, say for example a bacteria, virus, etc., is normally suspected only when the pathogen has produced enough disease symptoms. So here, say for example, you have a sore throat. So sore throat doesn't mean that the bacteria entered into your throat and within a second you developed sore throat. No. It will take 2 days or 3 days for that bacteria to completely occupy your throat and it to produce any symptom. So by the time the symptom is produced, already the bacteria would have resided in your throat for 2 to 3 days, right? So that is why the time taken for diagnosis of a particular disease will take time. But in the case of these molecular diagnostic, very soon itself you can detect the diseases. So by this time, the concentration of the pathogen or the number of the pathogen would have already become very high in the body. The best example that I can take off is urinary tract infections. It is not that all will have urinary tract infection symptoms. Some people, they will not at all develop symptoms, but by that time it would have caused a lot of damage in the body unknowingly because symptoms would have not developed. So at that time, it is always better to go in for early diagnosis techniques. So however, very low concentration of bacteria or virus at this time of when the symptoms of disease are not yet visible can be detected by using this particular molecular diagnosis technique, which is called as PCR. That is by amplification of the nucleic acid, wherein the sample can be taken, DNA can be extracted, that DNA can be amplified and then they can look into it or find out the disease. So PCR is one of the most routinely used or it has become a everyday use for the detection of HIV in suspected AIDS patients. Again here, AIDS, as we had studied in some of the earlier sessions we had studied, right? AIDS to develop symptoms itself, it will take many months to gather. So until then, the virus would have spread to the entire body and caused a lot of havoc there. So that is why instead of going and waiting for the conventional methods, using PCR technique, HIV can be detected before a person suffers from a symptom itself. So now and then, be it a mother or a couple, is suggested, or before going in for an operation, they ask us to go in for this particular test, right? Or before blood transfusion. So that is when they will not use conventional method. They'll go in for the molecular diagnosis method such as the PCR. And this PCR is used to detect mutations in the genes of the suspected cancer patients as well. Cancer also, it will be there in the body 5 years earlier itself, but we will not know about it. Why? Because we would have not developed any symptom. By the time we develop symptom, the cancer would be either in the third or the fourth stage, right, where it cannot be cured. So that is also the time when we can use this PC, polymerase chain reaction mechanism, or molecular diagnosis method to detect any mutation in the gene, so that early detection of cancer can be done. And this PCR is one of the powerful technique to identify many other genetic disorders as well. So this is a simple PCR machine wherein the samples are loaded and the test is done on this. So next is recombinant DNA technology. How recombinant DNA technology can be used to detect a particular disease. So a single-stranded DNA or RNA is tagged with a radioactive molecule which is called as a probe. So this radioactive molecule is allowed to hybridize with the complementary DNA strand. So what is done in recombinant DNA technology? If you remember, do you know that once the fragments are cut into small bits, we go in for a method called as polyacrylamide gel electrophoresis, right? That is gel electrophoresis, wherein the fragments will be separated. So for those fragments, probes can be designed. So those probes which are capable of emitting radioactive radiations. So these probes, what it will do is, when it will go and hybridize itself with or complement itself with one of the DNA strand on the gel, it will start to illuminate. So that time we can find out whether there is any problem in that particular strand or not. So therefore, very easily the clone having the mutated gene will hence not appear on the photographic plate. So how it is done, I'll show you. Say for example, all the DNA fragments are there on the gel slab like this. So this is single stranded DNA, right? It got separated. So say for example, this is ATG. Now I will design a probe. What probe should I design? I should design a probe that is complementary to this. What will I design the probe? I'll design the probe T. This is TAC. Now this TAC will go and bind here, right? This will be radioactive. So if it goes and binds here, it is radioactive. So if it goes and binds here, what will happen? When we look at it under the UV light, it will illuminate. It will release a light. So saying that, okay, this particular strand of DNA is normal. Now say for example, I've created this probe, but this gene fragment had got mutated. It might have been TTG. Now will this go and bind here? No. Why? Because T doesn't complement T. T always complements only A. So when it doesn't go and bind here, will this get eliminated? It will not get eliminated. Then I can understand, okay, something is wrong with this portion of the gene, and we can conduct studies on that particular portion and find out what is the actual problem, which protein it is producing, and therefore what is the disease there. That is how we can find out about the mutated gene under recombinant DNA technology. Next is ELISA. So that is enzyme-linked immunosorbent assay, ELISA. So ELISA is based on the principle of antigen-antibody interaction. So antigen is nothing but the foreign body. Foreign body in the sense it can be a virus, it can be a bacteria, it can be anything. A pollen from outside, anything, that is a foreign body. What is the antibody? Anything that is produced in our body to fight against a foreign body is an antibody. So the interaction between the antigen and antibody studies is usually done under ELISA. So here infection by the pathogen can be detected by the presence of antigens. Say for example, they will contain some antigens such as proteins, glycoproteins, etc. Or by detecting the antibodies synthesized against that particular pathogen. So, here we will find out an antibody has been produced in the blood, be it a B lymphocyte or a T lymphocyte would have been produced. That B lymphocyte or T lymphocyte that is capable of killing that particular antigen or subsiding that particular antigen. So, therefore, we can find out when that particular antibody is produced, we can find out that, okay, this person has been diseased by this particular organism and a proper treatment can be given. That is what is done in enzyme-linked immunosorbent assay, which is usually used for detecting AIDS at an early stage, HIV or AIDS at an early stage. So, this was about this particular session wherein we studied about how biotechnology is used in medical field, and we studied how biotechnology is used in early detection of diseases by using molecular diagnosis, and also how biotechnology is used to produce pharmaceutical products on a larger scale, wherein we took the example of the production of insulin, that is through genetic engineering. So, in the next session, we shall study about transgenic animals, how biotechnological approaches are used in the production of transgenic animals, and what are the different ethical issues that have arose because of this particular advancement in biotechnology. So, this is a very interesting topic. So, we shall meet again in the next coming session. Thank you.